We are going to begin with part 1 of carbohydrate metabolism. Today we're going to simplify a high-yield topic from this topic, which is the entry of glucose into cells. This is key for understanding carbohydrate metabolism and shows up often in both MCQs and SEQs. So let's break it down with real logic, exam cues, and quick recall pauses. Grab your notes and let's lock this in for good. Glucose is the body's main fuel, but it can't just walk into cells. It's polar and water-soluble, so it needs help to cross the lipid bilayer. That's where glucose transport systems come in, and understanding them is essential for both physiology and pathology. There are two main types of glucose transport: facilitated diffusion and active transport. Let's begin with facilitated diffusion via GLUT transporters. This is a passive process, meaning it doesn't require energy. Glucose moves down its concentration gradient through specific glucose transporters called GLUTs. First up is GLUT1, found in red blood cells, the brain, and the cornea. Its role is basal glucose uptake. A deficiency in GLUT1 can lead to GLUT1 deficiency syndrome, causing seizures and developmental delays. A typical MCQ might ask, which GLUT is responsible for glucose transport in RBCs? Next is GLUT2, found in the liver, kidneys, pancreatic beta cells, and the small intestine. It's bidirectional, allowing glucose to move in and out of cells. Its job is to help regulate blood glucose, especially after meals. A common SEQ might ask you to explain how GLUT2 plays a role in glucose sensing by pancreatic beta cells. Pause and think. Which GLUT is both for uptake and release? GLUT3 is the major glucose transporter in neurons. It has high affinity, which means it ensures the brain receives glucose even when blood levels are low. In MCQs, you might be asked, which GLUT has high affinity and is found in neurons? GLUT4 is found in skeletal muscle and adipose tissue. It is insulin-dependent, meaning it gets translocated to the cell membrane only when insulin is present. This is a crucial point in diabetes, especially type 2, where GLUT4 response is impaired, leading to reduced glucose uptake. A high-yield SEQ might ask, describe the role of GLUT4 in insulin-mediated glucose uptake. Pause and think. Why does insulin increase glucose uptake in muscles but not in neurons? GLUT5 is a bit different. It transports fructose, not glucose. It's found in the small intestine and sperm. MCQ alert. Which GLUT specifically transports fructose? Now let's talk about the second transport mechanism. Active transport, specifically sodium-dependent glucose transporters, or SGLTs. These work against the glucose gradient, meaning they require energy indirectly via the sodium gradient. SGLT1 is found in the small intestine and renal tubules. It co-transports one glucose molecule along with two sodium ions into the cell. Mutations in SGLT1 cause glucose-galactose malabsorption, which can lead to severe diarrhea in infants. In SEQs, you may be asked to explain the SGLT1 mechanism and its role in intestinal glucose absorption. SGLT2 is found in the proximal renal tubule. It's responsible for glucose reabsorption from urine. Clinically, SGLT2 inhibitors are used in type 2 diabetes to reduce blood glucose by promoting glucose loss through urine. A potential MCQ could be, what is the function of SGLT2 in the kidney? Pause and think. Why do SGLT2 inhibitors cause glycosuria? For your quick exam revision, in SEQs, always follow this structure: transport type, transporter, location, function, and clinical link. For MCQs, focus on which GLUT is found where, which are insulin-dependent, which transporter handles fructose, and what causes glucose-galactose malabsorption. Your turn now. Which glucose transporter do you always mix up? GLUT2 or GLUT4? Write it down or drop it in the comments. Our next topic is hormones that regulate carbohydrate metabolism, the key players in maintaining our blood sugar levels and energy balance. The major hormones involved include insulin, glucagon, epinephrine, cortisol, growth hormone, and thyroid hormones. Each of these has a distinct role, either promoting storage or release of glucose depending on the body's needs. Insulin, secreted by the beta cells of the pancreas, is the main anabolic hormone. It lowers blood glucose by promoting glucose uptake into cells, especially in muscle and fat, and by stimulating glycogenesis, lipogenesis, and glycolysis while inhibiting gluconeogenesis. On the other hand, glucagon, secreted by alpha cells, is catabolic. It raises blood glucose levels by stimulating glycogenolysis, gluconeogenesis, and lipolysis, especially during fasting states. Epinephrine, released from the adrenal medulla during stress or hypoglycemia, boosts blood glucose by rapidly stimulating glycogenolysis in liver and muscle and inhibiting insulin secretion. Cortisol, a glucocorticoid from the adrenal cortex, supports long-term glucose availability by enhancing protein breakdown for gluconeogenesis, decreasing glucose uptake in tissues, and maintaining blood glucose during prolonged stress. Growth hormone from the anterior pituitary reduces glucose uptake by cells and increases lipolysis, helping spare glucose for the brain. Lastly, thyroid hormones like T3 and T4 increase basal metabolic rate and enhance the overall rate of carbohydrate metabolism. Now, what happens when these hormones are deficient or in excess? Insulin deficiency, as seen in type 1 diabetes mellitus, leads to hyperglycemia, ketosis, and muscle wasting. Excess insulin, on the other hand, can cause hypoglycemia, leading to confusion, seizures, or even coma. Glucagon deficiency is rare but would impair gluconeogenesis and cause hypoglycemia, while excess contributes to hyperglycemia, as seen in glucagon-secreting tumors. Epinephrine deficiency reduces stress response and glycogen breakdown, whereas excess increases blood glucose and can worsen diabetes. Cortisol deficiency, like in Addison's disease, leads to hypoglycemia, fatigue, and weakness. In contrast, excess cortisol, as in Cushing's syndrome, causes hyperglycemia, muscle wasting, and insulin resistance. Growth hormone deficiency may cause hypoglycemia in children, while its excess, especially in adults, contributes to insulin resistance. Thyroid hormone deficiency, or hypothyroidism, slows down carbohydrate metabolism, leading to fatigue and weight gain, while excess, as in hyperthyroidism, increases glucose absorption and insulin demand. In short, hormonal balance is essential to maintain blood sugar homeostasis, and disturbances in these hormone levels can result in serious metabolic consequences. Now let's break down one of the most commonly tested and misunderstood topics in biochemistry, glycolysis. Yes, the UHS absolutely loves it. So if you're asked questions like describe the glycolytic pathway along with its regulation and significance, or calculate ATP yield in aerobic versus anaerobic conditions, this is what you need to remember. Glycolysis is a catabolic pathway that converts one molecule of glucose into 2 molecules of pyruvate. This process occurs in the cytoplasm and produces a net gain of ATP and NADH without requiring oxygen. Why is it important? Because it provides rapid energy and supplies essential intermediates for fatty acid, amino acid, and nucleotide synthesis. Now, the 3 irreversible steps of glycolysis are extremely important. These are the highly regulated one-way reactions that keep the process moving forward. 1. First, the conversion of glucose to glucose 6-phosphate catalyzed by hexokinase or glucokinase. Hexokinase is inhibited by its product, while glucokinase is activated by insulin, especially in the liver. 2. Second, the rate-limiting step catalyzed by phosphofructokinase 1 or PFK1, which converts fructose 6-phosphate to fructose 1,6-bisphosphate. PFK1 is activated by AMP and fructose 2,6-bisphosphate and inhibited by ATP and citrate. 3. And finally, pyruvate kinase catalyzes the conversion of phosphenylpyruvate, PEP, to pyruvate. It is activated by fructose 1,6-bisphosphate and inhibited by ATP and alanine. That leads us to a very high yield comparison, hexokinase versus glucokinase. Hexokinase is found in most tissues, has a low Km, meaning high affinity, and is inhibited by its product, glucose 6-phosphate. It works efficiently even at low glucose levels. Glucokinase, on the other hand, is found in the liver and pancreatic beta cells. It has a high Km, low affinity, and a high Vmax, meaning it works best when glucose is abundant. It is not inhibited by glucose 6-phosphate and acts more like a glucose sensor. A common MCQ tests which enzyme works during fasting versus feeding states. Make sure you know the difference. During the feeding state, also known as the postprandial state, which occurs within about 0 to 4 hours after a meal, the dominant hormone is insulin. The body's primary goal in this phase is to store nutrients and build reserves. Metabolically, several processes are upregulated. Glycolysis increases, converting glucose to ATP for immediate energy. Glycogenesis is activated, storing excess glucose as glycogen in the liver and muscles. Lipogenesis begins, turning extra glucose into fatty acids for long-term storage. And protein synthesis ramps up. Since blood glucose levels are high, most tissues use glucose as their main energy source. Key enzymes in this state include glucokinase in the liver, which becomes active under insulin and handles high glucose levels efficiently, and PFK1 and pyruvate kinase, both of which are also stimulated by insulin to keep glycolysis running smoothly. In contrast, the fasting state or post-absorptive state begins several hours after eating, typically between 4 to 16 hours. Here, glucagon becomes the dominant hormone, supported by cortisol and epinephrine in prolonged fasting. The body now shifts its goal to maintaining blood glucose levels, especially to fuel vital organs like the brain and red blood cells. To achieve this, gluconeogenesis increases to make new glucose, glycogenolysis breaks down stored glycogen into glucose, lipolysis mobilizes fat for energy, and in later stages, ketogenesis kicks in to provide ketone bodies as an alternative fuel. During fasting, hexokinase remains active across tissues due to its low Km, allowing it to function well even when glucose is scarce. However, PFK1 and pyruvate kinase are inhibited by glucagon, typically through phosphorylation, which helps reduce glycolytic activity and conserve glucose. And here's a simple way to remember it. Insulin in, glucagon gone. When you eat, insulin dominates and the body stores and uses glucose. When you fast, glucagon takes over to save glucose and generate more when needed. Now, let's talk about the inhibitors of glycolysis, which are frequently asked in exams. Fluoride inhibits enolase and is commonly added to lab tubes to preserve glucose levels. Arsenate inhibits glyceraldehyde 3-phosphate dehydrogenase, disrupting ATP production. Moving on to aerobic versus anaerobic glycolysis. In aerobic conditions, pyruvate is converted to acetyl-CoA and enters the TCA cycle. In anaerobic conditions, like in red blood cells or exercising muscles, pyruvate is converted to lactate. Aerobic glycolysis yields 7 ATP per glucose, 2 directly and 5 from NADH. Anaerobic glycolysis yields only 2 ATP since NADH isn't used in oxidative phosphorylation. This contrast is important for understanding metabolic adaptations and is a common MCQ. Now here's a clinical link: pyruvate kinase deficiency. This enzyme catalyzes the last step of glycolysis, producing ATP. In red blood cells, which lack mitochondria, glycolysis is their only energy source. A deficiency here leads to reduced ATP, causing failure of the Na+, K+ pump, cell swelling, and ultimately hemolytic anemia. This condition is often tested in SEQs, so remember, ATP depletion leads to membrane fragility and hemolysis. Now let's compare substrate level and oxidative phosphorylation. Substrate level phosphorylation occurs directly in the cytoplasm during glycolysis, requires no oxygen, and involves enzymes like pyruvate kinase and phosphoglycerate kinase. In contrast, oxidative phosphorylation happens in the mitochondria, requires oxygen, and uses the electron transport chain and ATP synthase to generate ATP. Both types are vital for energy production, and their differences are often tested in theory and MCQs. Let's wrap up with a rapid recap for your SEQ preparation. Glycolysis occurs in the cytoplasm, converting glucose to pyruvate. It yields 7 ATP aerobically and 2 ATP anaerobically. Key enzymes include hexokinase or glucokinase, PFK1, and pyruvate kinase. It's regulated both allosterically and hormonally. Insulin activates it, while glucagon inhibits it. Clinically, Pyruvate kinase deficiency causes hemolytic anemia. ATP is generated via substrate-level phosphorylation, and glycolysis is targeted by several inhibitors like fluoride and arsenate. Don't forget your comparisons: aerobic versus anaerobic glycolysis and hexokinase versus glucokinase. And finally, quiz yourself. What is the main regulatory enzyme of glycolysis, and what activates or inhibits it? What are the two steps where substrate-level phosphorylation occurs? And what causes hemolysis in pyruvate kinase deficiency? Say it aloud, write it down, or drop your answers in the comments. Whatever helps you lock it in before your next exam.